Good evening everyone, and welcome to our annual Koadlow Lecture. It gives me great pleasure to have Associate Professor Megan Munsie here today talking about stem cell therapy and their use or misuse in the management of musculoskeletal conditions. For those not familiar with MOVE, we're a consumer organisation supporting people with musculoskeletal conditions such as chronic back pain, arthritis, osteoporosis, gout, ankylosing spondylitis, fibromyalgia, and many others. There are 150 conditions which fall under the umbrella. And we provide support through our helpline, information resources, lectures, webinars, and as well as providing advocacy to give people a voice who have this condition. Before we start, I'd like to acknowledge the Koadlow family whose generosity has made these lectures possible. Just to give you a bit of history, almost 50 years ago, a passionate rheumatologist, Les Koadlow, started this organisation for newly diagnosed patients to find support regarding their condition. A stalwart believer in the benefit of real people supporting real people, he set up Arthritis Victoria, which is now MOVE, as a place for those living with musculoskeletal conditions to be informed and get supported. The Koadlow Lecture helps us inform people about research, new treatments, and the strength of this research around these, so that people are fully aware of the benefits, or lack thereof, and the risks, so that they can be informed and make informed choices about their care. With that, now I'm very pleased to introduce to you our guest speaker, Associate Professor Megan Munsie, who is based at the University of Melbourne, where she heads the Education, Ethics, Law and Community Awareness Unit at Stem Cells Australia, and she is also the Deputy Director at the newly established Centre for Stem Cell Systems. Over the course of her 20-year career in stem cell science, Megan has developed expertise in stem cell research and its clinical translation, and has made substantial contribution to the development of policy at a domestic and international level. Megan Munsie is also a great supporter of MOVE, assisting us in the development of our stem cell consumer resource, as well, of course, agreeing to be our guest speaker at this lecture. So please join me in giving her a warm welcome.
Thank you so much, Ornella, and thank you all for coming out, braving the weather and joining us tonight. I just want to reiterate that I really would love tonight to be quite informal. So while I'll go through and give you some information, I'm really looking forward to the question and answer session, because I think it's only when we really start discussing and talking about, particularly about the science and what it involves, that we'll really get a firmer understanding of its potential, but also its limitations. So I want to kind of really emphasise today where we're at in the stem cell field. And I'm sure you've seen headlines like these. And it almost feels like every week there's a new news story about stem cells. And what I find really frustrating as a scientist is that the headlines are often quite disconnected from the reality of the science. It might be in the final paragraph of a lengthy article where you finally find out that the big discovery has actually been made in rats or in mice or maybe even a laboratory dish, and yet to even go anywhere near humans. And it's the potential that's being spoken about in the headline. So I find this a little bit concerning. I mean, it's exciting. I'm excited as a stem cell scientist, and I suspect I have contributed to some extent to hype over the years. But I hope that my excitement is a bit reality-based. So before we go any further, let's talk about what stem cells actually are. There's been some survey work where researchers have asked people in the community, Australians in the community, about whether they're aware of stem cells. And there was a survey a couple of years ago where they were asked to kind of comment and rank different areas of biotechnology, vaccine development, genetic modification, stem cell science. And they were asked whether they were aware, whether they saw any benefit, or had any, I suppose, concerns about risks. And what I found fascinating in the survey results was that stem cell science was seen as being the most well recognised, the area of biotechnology that had the most anticipated benefit of all of those things looked at, but also the area that had the lowest risk. So high expectations about this field. So what are stem cells? So this is a little schematic that I like to use. A cell doesn't kind of look like that. That's a very, stylized version. What I'm talking about are the cells in our body, and stem cells are a particular cell in our body. So when we look if we look in with a microscope at the tissues and the tissues in an organ in our body, they're composed of cells. And there's about 200 different types of cells in our body. They usually share the same attribute in that they have a nucleus. And in this image, it's a pretty purple colour, and it's surrounded by cytoplasm, which is the blue colour. But depending on its function and where it works and how it works in the body, that cytoplasm can be long and thin, it can be highly interactive with its neighbouring cell, or it can be like this little spidery one down the bottom here, which is a nerve cell, have a body that's quite tight with lots of long spindles radiating out of it. So the cells morphologically, they look different and they have different attributes. What a stem cell does is it has a nucleus, like most of those cells in the body do, where the DNA is. But it has these unique biological properties that means that a stem cell can make itself again, so it can kind of replace itself. But it can also, given the right conditions, make new specialised types of cells. So whilst this arrow, it's called differentiation, that process, which basically means development. So that arrow implies it's just like from A to B, but it's actually multiple steps along that pathway to get to B. But what makes stem cells so unique is that they can make these other cell types. So they're kind of a primitive cell that, given the right coaxing and circumstances, can grow. Now, the other thing I want to impart tonight is that there are many different types of stem cells. So whilst they all share these basic properties, I like to think of two kind of broad categories of stem cells based on where they come from. The first category is what I would call tissue stem cells, or you might have heard of adult stem cells before. And these are stem cells that you isolate from tissue or from organs within the body. And here in this schematic, we can see examples of acknowledging that there's stem cells in skin, that there's stem cells in muscle, including heart muscle, that there's stem cells in our gut, that there's, and I have my glasses back on to see the rest of it, sorry, that there's stem cells in skeletal muscle, and that there's stem cells in bone marrow, that's in our long bones, for example, that makes our red blood cells and our white blood cells and our platelets. And really, we've known about those stem cells in these organs that can regenerate, like gut, like blood, like skin, for a long time. Because we know if you cut yourself, your skin repairs. In our gut, we shed hundreds of cells, but the lining of the gut is restored every day to compensate. Similarly, in our bone marrow, the stem cells are there to maintain our health. If we don't have healthy blood stem cells, and perhaps there's something wrong with them, you can develop leukemia. So these are broadly called tissue or adult stem cells. I don't like the term adult stem cells because, of course, we also use stem cells derived from fetal tissue in research. So I prefer tissue. Whilst these cells have that ability to make another primitive cell or to differentiate, these types of stem cells usually can only grow into the cells from the organ in which they're generated. So a blood or bone marrow stem cell that can usually only make red blood cells, white blood cells and platelets, so cells of the blood system. It can't usually make brain cells. Equally, if we take a cell from the gut, a stem cell from the gut, it can only usually make those cells that reside in the gut. It can't make brain or maybe skeletal muscle. So the other type of stem cells that get a lot of coverage, and here I'm thinking back, and you might remember some of the debate when John Howard was Prime Minister around the use of human embryos in research. So in the late 90s, colleagues in the US from Jamie Thompson's lab identified that you could take an embryo that was made in IVF, and I'll come back to that in a minute, but made in an IVF clinic, and you can take cells from that embryo and make stem cells. So these are called human embryonic or ES, human embryonic stem cells or ES cells. And this little illustration is just showing the different stages. So this is not fetal research. And in Australia, since 2002, we've been allowed to use excess donated IVF embryos to make these stem cells. And why? And there are lots of, whilst we can do that, there's lots of requirements for researchers to meet. It's permitted, but highly regulated. We have to apply for a license, for example, to do research, which I think is right. I think there is a lot of contentious issues around using embryos, and perhaps in the question time we can talk about some of those. But I wanted to make a point that these type of stem cells, why the research community was so eager to use them, is they're quite different from those adult or tissue stem cells I showed before. Rather than only being able to make one type of mature cell, like start on a pathway to make a skeletal muscle cell or to make cartilage, you can actually coax these cells into forming lots of different types of cells. So that one type of stem cell can actually make all the cells in the body, given the right circumstances. Now, it's not as simple as that, and over the last couple of decades, we're getting better and better and better at directing its fate, but it's still not absolute. But the potential is, from the scientific perspective, mind-boggling, because it means that you could have almost a universal source of cells that, given the right circumstance, we can use. So this is the basis of some of the excitement and also the controversy that you might remember reading about some sort of 17, 16 years or so ago. The other discovery that was made in 2006 in mouse and in 2007 in humans was that you don't actually have to use an embryo to make these stem cells. These embryonic stem cells, the first ones I showed you, are called pluripotent stem cells because of this ability to make, pluri is just another way of saying many, so many different, a potential to make many different cell types. These iPS cells that Shinya Yamanaka, a Japanese researcher, and his colleagues identified, you can actually create from a skin scraping. So you can take a skin cell, or indeed any other body cell, and you can pull it back down that highway of differentiation back to a primitive pluripotent stem cell. Now, this discovery was made, as I said, almost 10 years ago, and Shinya was awarded jointly the Nobel Prize for Physiology and Medicine in 2011 from this discovery. So a relatively rapid acknowledgement of the contribution in this field. And again, these cells can be made in the laboratory, and then they have a very similar ability to make lots of different cell types. So this was a marvellous discovery because it meant we didn't have to use human embryos, but more importantly, we could take a cell from a patient with a condition, and potentially we could make cells that could match that patient, and if we use them for transplantation, they wouldn't be rejected. But I think more importantly, we can understand disease. We've got a new tool now to understand what happens, what goes wrong in the body in conditions like motor neuron disease, for example. What goes, and conditions where it's difficult to perhaps identify or get a sample from a patient. We can now mimic that in a laboratory dish. A lot of medical research relies on the use of animals and mimicking human disease in animal models. This means that we've got a new way of tackling some of these difficult, challenging questions about what goes wrong in disease and what goes wrong in development sometimes. So this is a new form of stem cells, and a lot of groups are doing work around, are using these stem cells around the world. And again, we can talk a bit more about them later perhaps. But what makes stem cell research or stem cells so valuable for research? So this schematic again is just acknowledging that there's two types of stem cells, but there's many applications. I want to focus initially on research. I've just given you some examples about how we use stem cells in research to understand disease. But we can also use them to understand normal development. And colleagues are taking pluripotent stem cells and tissue stem cells for that matter, and they're trying to mimic organ formation and organ development. So colleagues at University of Melbourne are doing some lovely work looking at cancer of the gut. And they're taking small biopsies from the lining of the gut, and then they're making little mini guts in a dish. They're called organoids, just to make things a little more complicated, I suppose. But we can make them, and they replicate those cells that form the lining of the gut. And by doing that, we can interrogate and look at cell interaction and how infections occur or how cancer develops, for example. Other colleagues are taking the pluripotent stem cells, either embryonic or these iPS cells, and they're coaxing them down, trying to mimic what happens in fetal development, looking at how to make an organ like the kidney. So we can't yet make little tiny miniaturized kidneys, but what we can do is mimic that development. And that tells us so much about what's going on, things that we would never know without this sort of new insight. We'll certainly never know with human cells without this new tool. The other application of stem cells is in drugs. And here I'm not talking about a capsule that's got stem cells inside it that you might swallow. I'm talking about using stem cells in drug discovery and in toxicology studies to kind of add again to pharmacology and development of new drugs, where we can use stem cells and cells made from stem cells to screen for whether a promising drug is going to work. So again, it adds another dimension to medical research. But of course, I bet the thing you've been thinking about is cell therapy and how stem cells could be used in cell therapy. And of course, that's what I want to concentrate on today. And that's, I think, the growing area. But I just wanted to, again, emphasise that we are using stem cells all the time around the globe in basic research, and they're a really important part of basic medical research. The final comment to make here is that, of course, not one stem cell can do all of those things. And as a research community, Stem Cells Australia is an Australian Research Council funded initiative where we have about 300 researchers across the country working to look at these questions about how we can use stem cells in research, how we can develop new cell therapies. And we use a variety of stem cells. We look for the right type of stem cell for an application. So there's not sort of one stem cell fits all. Even though the ideological argument, particularly around the use of human embryos, was to say that adult stem cells are so much better than embryonic. We don't see that. We look for fit for purpose and what's going to accelerate and advance our research. When it comes to cellular therapies, the reality is that there are very few proven and effective stem cell treatments, unfortunately. I wish this wasn't the case. But I wanted to acknowledge that in the case of using blood or hemopoietic stem cells from bone marrow, we've been using them for over 40 years. In fact, we've been using cord blood now for decades. So cord blood is taken from the umbilical cord of a newborn, from the placenta following birth. And we've been using hemopoietic or blood stem cells, as I said, for decades. But we use them to treat diseases of the blood system. We don't use them to treat people who've had a stroke or someone who perhaps has muscular dystrophy. We use them to help people who have something wrong with their blood system or perhaps need to have some additional stem cells as a result of perhaps chemotherapy or radiation therapy. So a proven, accepted, but also highly researched area. We're still optimising how to do this better. So there is still a lot of active research being undertaken. Similarly with skin, I mentioned earlier that there are stem cells in skin, and for decades we've been exploiting that with skin grafts. And you might remember seeing some images, particularly after the Bali bombing, of how grafts and skin sprays, etc., were used to help people who'd been severely damaged following a burn. What we still can't do yet is replicate the complex organ that is skin. And whilst we can make a covering that can act as a barrier and stop infection and stop dehydration and loss of fluids through a wound, as you can see in the lower image, there's often a lot of scarring. The skin doesn't sweat. There's no nerves going into it. It doesn't sense temperature, can't regulate temperature. And obviously, for a child that's growing, that can also cause a problem if the skin graft can't stretch as the child grows. So again, there's a lot of work being done to optimise that, but it still relies on stem cells. It is being used in medicine at the moment. The final area I wanted to highlight was a form of blindness where the surface of the eye is damaged. And often it is actually an industrial burn or a splash to the eye. So otherwise the eye is fully functional. But something happens to the surface of the eye which results in scarring. There are rare congenital diseases where this can also occur. So the eye is otherwise fully functional, but the surface of the eye is damaged. And initiated by decades of research in Italy, scientists and clinicians have now developed a new way of addressing this problem, provided there's a ring of, start that sentence again. There is a region of stem cells in the eye, it's just outside the iris, called the limbal stem cell zone. What you can do, if that region isn't damaged, is you can take a small biopsy, put it on a scaffold, like you can see on the top. That's a little piece of membrane. You can put the little biopsy of tissue on that, and it will grow out. And it will grow out just like the normal surface of the eye. And what a surgeon can then do is cut away the damaged cornea and add back the clear cornea. So this is using the patient's own cells. It's recently had European government approval, and we have some fabulous researchers at the University of Sydney and University of New South Wales working on developing an Australian solution. And in fact, they won, do you remember the Young Inventors Award from a decade or so ago? They won that award for their work, and we'd really like to see that continued. They're actually looking at using a contact lens as the surface. So they effectively make a band-aid, making the cells grow over the contact lens, and then cut out the damaged part of the eye and then put that back over the eye. So that's an example of how some of the Australian researchers are extending the work that was started in Italy. So this is all of these examples are have been as a result of years of research and clinical practice. Beyond this, we should think about the application in terms of stem cell therapies as being experimental. And I think really should be in the realm of clinical research, where a patient is really only accessing these interventions if they're participating in a clinical research program, a clinical trial, for example. And this is a screen grab from the National Institutes of Health clinicaltrials.gov website, which is a database that has a registry of all clinical trials that are happening around the world. And you can search it on different conditions. And here you can see I've searched for osteoarthritis knee, and then sub-searched for stem cells. And the search shows me that at the moment there's 61 trials that are either underway, about to begin, or recently completed. And you probably can't read the small type here, but the type of stem cells that they're using range from a particular type of tissue stem cell called a mesenchymal or MSC cell that I might talk to you a little bit more about in a moment, that's been derived either from bone marrow or from fat. And they've also used Wharton's jelly or a part of the placenta to isolate these cells. So I just wanted to give you a taste for the work that is being done around the world at the moment. These clinical trials are being conducted in India, Jordan, China. And again, I've got a little bit more to say about this kind of database, but the take-home message here is that if we inserted motor neurone disease or cerebral palsy or any condition that you can think of, there is a clinical trial happening around the world. And just to introduce a little bit more about what a clinical trial is, I thought this schematic was quite helpful. And a clinical trial really should be the first time a proposed intervention, a proposed new treatment is trialled in humans. But it should only be done, as this schematic indicates, after several years, maybe decades, of laboratory research, of perhaps animal research, to show that this has promise, that this approach is feasible. A clinical trial will start at either phase 1 or phase 2, where really only a handful of patients, maybe a couple of dozen, up to 50 patients, are enrolled. And the objective here is to look at safety. It's the whole premise of that first do no harm. Does this new intervention put the patient's life or health at risk? That's a fundamental question. And whilst it's the main aim of the early phases, it is maintained even after decades of investigation into a new therapy, where you continue to monitor patient progress and report on a treatment. The latter phases of the clinical trial, phase 3 or phase 4, as you can see in the schematic, involve a lot more patients, and their design is to look at safety, but of course to look to see whether the intervention works. And at the later stage, to see if the intervention works against or in comparison to another type of therapy. So what's the current benchmark, I suppose? What's being used? Is this better or worse? So it's a way of testing it. I don't have any hard and fast data on this, but I fear from talking to a lot of people in the community that they often think of clinical trials as being a shortcut to a proven treatment, because usually you don't have to pay to participate in a clinical trial. But I just want to put it to you that I think this is research. It's really valuable research. It's still research. We don't have all the answers. And in fact, unfortunately, sometimes things do go wrong in clinical trials. So there is a consent process, and there's a lot of review and reflection about, justification for a clinical trial. And I think that's right. These are experiments on people. So how could stem cells help in cellular therapy? So I put up a couple of points here around how stem cells could benefit musculoskeletal problems and bone and joint issues. So obviously, if we think back to the example of of blood stem cells and that idea of someone who has leukemia, where there's something wrong with a particular cell in your blood, that if you only replace the stem cells, you could restore function. Why couldn't it work like that with joints? If there's something wrong with the cartilage, why not just put more cartilage in there? Well, maybe stem cells could be a source of new cartilage, and or a source of new bone, or a source of new muscle. And that's definitely, in laboratory research, we can take particularly those pluripotent stem cells and make large numbers of cartilage cells or bone cells or muscle cells. So it's feasible from that perspective. But the challenge will be how to get those cells grafted into the organ where they're needed. So how do you deliver them? So that might be possible. The second dot point is around whether the cells, the stem cells or cells made from stem cells, could be used to perhaps not graft, not embed and implant, but to talk to the cells that are already in the joint, for example, to coax them out of their inactive phase and to say, come on guys, can't you repair yourselves? Some kind of regenerative property. So there's evidence that certain stem cells have an ability to talk to other cells and perhaps affect that balance between active repair and inactive repair. Because whilst there's some organs in the body that repair spontaneously, like gut, like skin, other organs don't do that, even though there's stem cells there. So that would be a really interesting way of using stem cells. So it wouldn't involve the cell going in there and engrafting, but it would be talking to other cells that are in the joint, or the muscle, for example. And maybe while it's there, it could prevent or arrest further degeneration. So that's also been proposed. The final point is that certain types of stem cells, like those MSCs or mesenchymal stem cells I mentioned a moment ago, have been shown in laboratory studies to be able to sort of dampen down the immune system, and also in animal studies. So could they be a way of moderating or reducing inflammation and therefore pain? So that if they were able to do these things and be kind of like little cellular warriors to go in there and address the damage or disease, that would be another way that stem cells could be used. But of course there's many questions that we don't know yet around how stem cells could work and the limitations around these approaches. So those 4 points I've raised, I think you could say they're hypothetical justifications for doing cellular or stem cell-based therapies. Some of the raw questions for me are around what stem cell or cell made from stem cell is required for a certain condition. It's not likely that one stem cell type will fit and be able to do all of these things. If you want cartilage, you want a cell that is going to either make new cartilage or has that property itself. You don't want it growing into a nerve in your knee. You need to be able to put back sufficient numbers to cause benefit, but not cause harm. So how many is that? What condition and when in the progress of that condition is it likely to benefit? So if we look at osteoarthritis and the different grades of osteoarthritis, and perhaps we could use these MSC cells, for example, are they going to be effective early in preventing damage? Are they going to be effective when there's still some residual cartilage there? Or is it too late to use them when someone's in stage 4 and there's no cartilage bone on bone? We don't know these questions yet, but they're really important and can only be addressed through well-designed clinical trials. Otherwise, we'll still be pondering these questions. Another issue that's often raised are the patient's own cells inherently better than using donor? Do a patient's stem cells age like we all age? And are we therefore better off using cord blood or cells made from cord blood? Or does that not matter? Maybe it depends on the condition. How are the cells prepared, by whom, and how are they delivered? So if we think about a joint, there's a lot of knowledge around how to deliver an injection into a joint. It's an isolated area. You could use perhaps radio guided delivery to make sure that the cells get into the joint. But what about bone? What about muscle? If we put these cells into a vein, will they get there? Or will they just get caught in the lung as they're delivered? So how they're put in is important. How they're prepared is also important. As soon as you take a cell out of the body, you're effectively changing it. So you could inadvertently introduce a contaminant, like a bacteria or a virus, even though you're using the patient's own cells. If you're using a contaminated reagent, for example, if you keep the cells in the laboratory over time, so maybe you want to expand and grow lots of stem cells or lots of different types of cells from a stem cell, are we sure that there's been no chromosomal damage? That you're not putting back a cell that's going to have an increased risk of growing into a tumour? And the other thing I was going to raise is that I've often been interested, particularly in joint applications, is whether we actually need cells at all. What's the therapeutic agent? What's the mechanism that's involved here? Does it rely on cells going in, or is the factors secreted or produced by the cells? And could we collect those and use those as an application? So I think there's many uses of cells and questions that need to be answered, again, in clinical trials. And finally, as I mentioned, the sort of culmination of clinical research should really be, does this new approach work better than what's already available? But of course, if you look at the newspaper, particularly the sports section, you might see a slightly different story. So here's some grabs of prominent sports players who've had stem cell treatment. Rafael Nadal for a shoulder injury, I understand. Adam Goodes for knee. And various football players for different types of sports and joint problems. And a very famous ice hockey player from Canada, Gordie Howe, who had a stem cell treatment for stroke that got an enormous amount of coverage. These, as I mentioned, these articles are often in the sports section that have a lot of hype associated with their treatment, around the stem cell treatment, without a lot of, I suppose, justification or analysis. It's just almost implied that if they're doing it, as this headline shows, no longer for highly paid athletes, you can come now and get stem cell treatments. So for people who are facing chronic pain, limitations in mobility, seeing articles like this inspire further research. And of course, if you hop online and do Dr. Google, which we all do, you'll come across ads like these. And I'm not sure whether the first one even uses stem cells. That one, just to read it to you, is knee injury is no fun, get rid of knee pain fast. I'm not sure whether they use stem cells. But the next one is pretty obvious, stem cells for knee. Osteoarthritis treatment in Sydney, it's an alternative to joint replacement, 600 patients plus treated, 90% success. The third one, you've heard of stem cells, experience it for arthritis. The one and only insured stem cell product, guaranteed for your safety, 99.9% free consultation. Now that's pretty alluring. Where is the evidence for these advertisements? And I suppose this is what I want to conclude on tonight, and is actually the focus of my research, is my concern around the manipulation of the excitement and the promise and the awareness of stem cell science into what appears to be a commercial industry that has no basis in evidence, but I think a big emphasis on profit. So this is a link that I got today from that last clinic that I showed. And of course you can't read all of this, but you can see the style of website. It looks pretty professional. It looks pretty welcoming. And in the text there, it describes an innovative technique using stem cells. And in fact, I've been following this company now for many years, and they've changed their website frequently. They allude to the fact that they reprogram or make pluripotent stem cells. So you remember I talked to you about iPS cells and how you can take a skin cell. In this case, they take a blood cell and allegedly they reprogram it back to a pluripotent cell and then put it back into your vein. Now, I did that in my mouse studies. Not into a vein, I put it back into actually the testis capsule. But I did that to show that it had tumorigenicity. It could make a tumour, it could grow into a cancer. That's one of the tests of pluripotency. So this I find quite concerning, that if they are doing this, the risk of changing that fate of that cell is really serious. When we use pluripotent stem cells and working towards a new cell therapy, we don't put back the stem cell. We put back a cell made from the stem cell, because we know that if you put back the raw stem cell, you run a risk of making a tumour. So the other thing I thought was fascinating about this screen grab is here, what are people saying? And I think the byline's something like real people, what real people are saying. And I just want to show you the quote from Juliana, who apparently is 66 years old from New South Wales. "Before I had my treatment, I was living with constant pain from my knees. I lacked energy, was feeling depressed. Less than 4 weeks post-treatment, all the pain is gone. I have energy misspelled. Then when I was in my 40s. I can now get out of bed easily, spend the whole day at the shops, walking around, and still have energy to play with my grandchildren. I wouldn't hesitate to recommend this treatment to anyone." Now, I really hope for this lady that's the case, and that she has had genuine and ongoing benefit. But it concerns me that the company are using a testimonial as evidence. This is not clinical research. This is not a clinical trial. It's a commercial activity masquerading itself as a clinical service. And unfortunately, this idea of selling stem cells and selling hope associated with stem cells is not new. We as a community, a scientific community, have been trying to grapple with this for over 10 years. We first started to see clinics opening up in China and India, the Bahamas and Mexico, about 2006, 2007. And I suppose you could say back then, we called it stem cell tourism because people were travelling overseas to pursue it, a bit like medical tourism. And you could probably argue that those countries had either no or limited regulations and oversight, but it was big business. And there were a handful. In fact, one of my colleagues from Canada did a study where I think they identified 13 clinics in 2008. Now there's hundreds of clinics that are operating. And what I wanted to show, this is a paper that was published last year, and this is a heat map of the world. And the darker the colour, the more clinics that are available selling stem cells. So this is not part of clinical research. These are clinics that are advertising direct to consumer, like the last one I showed you. And in fact, after this paper, another group published their work, their research, and identified in America there's over 300 clinics. Most of these clinics are claiming to use the patient's own cells. And they market them as safe because they're using the cells from you. What harm could it do? It's natural. And I would argue that it matters how the cells are processed, by whom, and how they're put back. It all changes the risk, and I'll talk a little bit about that more in a moment. Unfortunately, my 9 has dropped from the bottom, but in this paper they also identified that there were 19 clinics in Australia. Now, of course, that wasn't any news to me. We've been tracking this since about 2011 in Australia. I heard about the first, so I was active in this kind of observing and watching clinics overseas for many years, and then and taking a lot of patient inquiries. That's how I found out a lot about this issue. And I got my first inquiry from a gentleman who was concerned that his wife had been offered treatment for Parkinson's disease by a cosmetic surgeon in Sydney. And I thought, oh, goodness, can I... And I sort of talked to the gentleman about our concerns. And we have I'll tell you a bit more about our resources in a moment, but I told him about some of the things that we think he should be thinking about. And then, of course, I put the phone down and I rang the Therapeutic Goods Administration and my other colleagues to say, oh dear, I'm very concerned about this. And what I wasn't aware of was that there was a slight change in our regulation. So the Therapeutic Goods Administration are the government body that makes sure that our drugs and other therapeutics are safe, are tested, and protecting you effectively from harm. And what I didn't realise is that the TGA had introduced some new regulations around 2011 that were to perhaps increase the amount of regulation around the use of cells and other new and emerging technologies. But because they were concerned, and legitimately so, that maybe some current medical practices would be restricted, they put like a band-aid side bit of the legislation, an exclusion around certain things. Unfortunately, one of those things is any cell taken from a patient by a doctor. So what that means is that none of these practices have to gather evidence. They have a sort of an opt-out clause from having to go through the clinical trials network, which I still, to this day, find extraordinary. And as you can imagine, we've been petitioning the TGA and other organisations, so I don't think it's just the TGA that has regulatory oversight in this area, to address this issue. Because from that one clinic in Sydney, where this doctor was taking fat from liposuction, abdominal fat, and putting it back into the veins for people with all sorts of different conditions, we now have not 19, but in a paper we're about to publish, 77 clinics. Most of them are offering the treatment of osteoarthritis. And that's kind of why I'm here tonight and why I've been working with Ornella and Move, is that I think this is really alarming. And I'm not saying that there's no potential benefit in this therapy. It just needs to have some more research done before it's commercially and widely available, and some more follow-up. We need to make sure that the patients who've had it remain safe, we learn from their experience, not just keep treating them. So one of my key criticisms of these clinics is not only that they take money, in Australia the going rate's about $10,000 per treatment, but if it hasn't worked the first time, come back and try again. That's what concerns me. So if it was really a stem cell treatment, you'd expect it to engraft and do its job and work for a long period of time, not to have to keep coming back. So there's many other things we can talk about perhaps, but I wanted to show you another example of a clinic. Now this is in a suburban shopping centre in Broadbeach on the Gold Coast. Again, you probably can't read the small type, but this is a screen grab from their website, where they do definitely treat orthopaedic conditions, osteoarthritis, cartilage regrowth, tendon tears. But they also treat baldness, infertility, Parkinson's disease, dementia, multiple sclerosis, obstructive pulmonary disease and diabetes. There is no proven stem cell treatment for any of those. And yet this clinic's been operating for years. They do something similar where they take out the fat. They have a slightly different technique to prepare the cells. It's usually done as, I think, a same-day procedure. But I believe this clinic asks the patients to take quite expensive supplements as well as have the treatment. And in one couple that we've spoken to in our research that had a lung, the patient had a lung problem, she was told that it hadn't worked because she'd failed to keep up with her supplements. And there must be something wrong with her stem cells, which I think is blaming the patient. So what can we do about this? Well, as a community, the stem cell biologists, as I mentioned, have been long concerned about these exaggerated claims and tarnishing the work that we do. In fact, we're really concerned that not only will it give stem cells a bad name, but it also takes away the incentive for doing good clinical research, of really advancing the field. So I've just shown you that we have a lot of websites, including Stem Cells Australia, our website, but also one on the top right, a closer look at stem cells, that's sponsored by our International Society of Stem Cell Research, and it's been crafted to help get information out about stem cells to the general public. So we hope we're presenting information in a way that's user-friendly and helpful. Colleagues in Europe at Eurostemcell also have some marvellous fact sheets you can download that are basically, I think the title is, insert your condition of interest and stem cells and how can they help. And so you can get some really terrific information, because I think it matters what condition it is, what we're asking a stem cell to do for a condition. I don't subscribe to the magic bullet, stem cells will fix anything. And of course the marketing is implicit in the stuff I've shown you, that these clinics are kind of emphasising a stem cell has some kind of an amazing ability to float around the body and turn into whatever's required. Perhaps it does. It would be marvellous if it did. But I think I don't think we can really say that. And what we really need to have is a bespoke or tailored solution to a complex problem. The stem cell, cellular-based treatment we might develop for diabetes will be different to macular degeneration or to arthritis. It needs to be a little bit altered to be fit for purpose. So what we're trying to do in the information that we're providing is to challenge this hype, but also maintain hope. We're hopeful about stem cell research. We're hopeful about clinical trials. We want to see people participate in clinical trials. But while there's no requirement in Australia to do that with your own cells, there's very few clinical trials being done. So there's a real vacuum. And of course, we want to see legitimate clinical translation in clinical trials. Just to highlight some of the work that we've been doing with MOVE, hopefully you've got one of the fact sheets outside, but we were also delighted to contribute to the recent Update magazine, and we'll look to continue to do that because I think it's really important. The field's going to progress, and I hope to think that we can keep, as the field's evolving, we keep you informed. We also have patient information booklets, and there's various ones. Most of them are on our website, and these are, again, information booklets you can download. I think one of the best things about the Australian handbook and the International Society for Stem Cell Research handbook is that we have checklists and questions that you can ask someone who's offering you treatment to try to help you navigate whether it's legitimate or a bit suspicious, and also just get you more informed so you can have a really good conversation with your specialist. Because I think we all go online and we all search for information, but really we need to take that back to a doctor who knows us and knows our medical condition and can tell us about, discuss how this information you might glean is relevant or not for you and for your condition and the particular stage of your condition. What we also have done as a community is try to set up what we think are steps that need to be undertaken to advance the field. So these are guidelines that are published by the two major international societies, the International Society for Stem Cell Research, that I mentioned earlier, who will be coming to Melbourne next year, which is very exciting. They're having their annual meeting here. So hopefully you'll hear a bit more about stem cells in June next year. And also the International Society for Cellular Therapy. Both these organisations have published for their members, really, and for other professionals, things that should be required. What should be included in a consent form? What should a patient be told? What manufacturing standards should we require of someone who is exploring and developing a new cell-based therapy? So that's what we've been doing. And I also want to highlight that I think medical practitioners have a really big role to play here. And I've been delighted to start working with the Australasian College of Sports and Exercise Physicians. So these are specialist doctors. I'm just going to do a sidebar and say a lot of the clinics that I've been following, these stem cell commercial clinics that are selling unproven treatments outside clinical trials, they call themselves stem cell specialists. That is a nonsense term. There is no such thing. There's haematologists and there's physicians, there's oncologists, but there's no such thing as a stem cell specialist. So I'd just be wary of hearing that as well. Just because you're doing it doesn't necessarily mean you're good at it or informed. So what the college has done, which I think is fantastic, is they've taken a stance. So as a medical community, they're concerned about what their patients are reading and coming back and talking to them. So they have, and I'm just going to walk you through where to find it on the website. If you click the tab that's under resources, you'll come across their position statements, and the top one is their position on these mesenchymal or MSC cells. And there's a very detailed and quite dense, excellent review that was published in the British Journal of Sports Medicine in early 2016. But we're working with them to make a slightly easier to read version of that. So hopefully we'll be able to launch that later in the year, and we can write about it and let you guys know. But I think it's great that doctors are also, not just the scientists, but doctors are also saying, well, we'd like to be able to offer this to our patients, but there's some concerning aspects around where the field is at. And we need to have more evidence before we roll this out at scale and at a commercial level. We need more research, basically. So I just wanted to alert you to that and that resource and how you can get it. It's freely available on their website, and they'd be delighted if you were to download it. I'm going to conclude by talking a little bit about my research and what I do at the Centre for Stem Cell Systems at Melbourne University. My core interests are involved, as you might have gathered, around regulations. What do we need to have in place to enable safe and effective stem cell research and also the development of stem cell treatments? What should be required? And here's some papers that I've published recently with colleagues on that topic. The other core area of research is that we want to understand more about how the community view and experience stem cell research. So, in fact, I was reading this before here. I think it's share your story or tell your story. In fact, that sort of captures what we want. We want to hear people's story. And so I'm going to tell you a little bit about our research and how maybe you could be involved and what we've done to date. So we've just published a book on stem cell tourism. We've been working in this area for about 8 years. And as I mentioned earlier, we initially started to work in the area because we saw a large number of Australians travelling abroad to remote locations to have treatments that were highly invasive and carried substantial risk at huge cost. So I mentioned $10,000 before for joints. That's the going rate in Australia. But if you have a spinal cord injury, there's someone in India who'll treat you for $100,000. And in fact, I've met a gentleman who's had 3 treatments. And and then there's a lot more we can talk about that story, but I think it illustrates that high cost associated with these experimental interventions. And the high personal, the lengths that people will go to. I think it's completely reasonable that people explore these options, that they think about it. But what I'd like to do is to understand more about the decision making and whether we can better support that, aid that. So we've published a book. This was done with colleagues from Monash University, and we've published some papers. But let me just show you some of the results. So what we did in our main study is we interviewed about 50 Australians who were patients or their carers who had travelled or contemplated travelling abroad. Now, as I mentioned, most of these people were not suffering from osteoarthritis, but were suffering from very significant neurological diseases, particularly motor neuron disease, where they had a very limited, they were told that the disease was rapidly advancing, and they felt like they had limited time. We also spoke to several parents who had children with either birth defects or acquired defects, who felt like they had a window of opportunity that was very narrow to intervene while the child was still developing, to kind of stop the condition worsening. And when we spoke to people, this is all, of course, anonymous. It is conducted as part of a sociology study. And what was amazing was how willing people were to share their stories. The findings from the stories were really helpful for us because it challenges our assumptions. I talk to a lot of people about stem cells. I've formed strong opinions about the area, and I think I know what people are experiencing and how they're interpreting information they read. But the information that we get back from these interviews challenged my notion. I had always assumed that people in the situation would do their research, go online, but then they'd go to their specialist or their GP to talk about it before they made this expensive and challenging decision. But many people we spoke to said that the support from health professionals, and here I'm thinking predominantly around specialists, was very poor. In fact, to quote many people, they felt like they'd been through the ringer and that they were very frustrated, even angry with their health professional. They also, as I mentioned, felt like they had a narrow opportunity to try this, and they were frustrated that it wasn't here. Why is it happening in India but not Australia? Australia's fallen behind. It's outrageous. So there's a lot of anger about that. There was a lot of discussion around hope, and as you might have noticed, our book is subtitled The Economy of Hope. Because hope, it was a huge motivating factor, but very complex, in that people often define whether they meant realistic hope or, I suppose, something that was untangible. And the example I was going to share here was spinal cord injury. Many people we spoke to with spinal cord injury didn't necessarily expect to walk again. They might have secretly wished for that, but they didn't expect that. But what they wanted was bladder and bowel control, or maybe a little bit of sensation. So they just get a minor improvement. It was worth the 100,000 or whatever was being marketed to them. So they did talk about costs a lot. They talked about financial costs, and they talked about the cost of being away from their loved ones. So some of these treatments were offered over a long period of time. You couldn't just fly in and fly out. You had to stay there. And the costs associated with that were huge, but also you're disconnected from your support network and from your family. What I found astounding in these interviews, and most of the people we spoke to, the cells were delivered via lumbar puncture because they had neurological problems, and the doctors didn't just rely on the cells going into the blood and magically getting into the brain. They realised there was a blood-brain barrier. So to circumvent that, they injected these cells, and there was a mixture of types of cells that were used directly into fluid around the spine. Now that's a highly invasive procedure. It's a sterile area, and if you get that wrong, even with a little tiny infection, you get meningitis. So the idea of putting something that's prepared in a back room laboratory that maybe not purified, that also has the risk of growing into a tumour, is quite mind-boggling. Yet the people we spoke to, they almost casually mentioned lumbar puncture. Oh, I had a spinal tap. Yeah, it was put back in my child with spinal tap. And I found that really surprising. I would have thought that might have got picked up perhaps a little bit more. But risk to health wasn't mentioned. And I just wanted to share one quote from a woman who had a child with cerebral palsy who went to Germany. And as Lisa said in this quote, what's the worst that could happen? Nothing really. We could basically just lose our money. Now, a child died in that clinic. It was closed down not long after Lisa went there. Not because the cells grew into a tumour, but because the child had a seizure because the cells were directly injected into its brain and died as a complication. So these risks are rare but real. And I just wanted to finish on these two case studies. This was published this year in March, and I think it's a very chilling example of what can go wrong. 3 women who had macular degeneration were still able to drive. They were losing their vision, but they went to a clinic in Florida. They had their own stem cells put back into both eyes at the same time and were all rendered blind. Apparently there's a 4th woman who's now come forward as well. One of the women, at least, had gone onto that clinical trial registry and thought what she was participating in was a clinical trial. She felt like she'd done her homework. And she thought it was reasonable to pay, I don't know what the fee was, I think it was in the order of 30 or 40,000, but she thought it was reasonable to pay that to advance the field. So that's concerning. That's an example of a complication. There's another prominent example published the year before where a gentleman who had a stroke had the cells, fetal stem cells in this case, injected into his spine through a spinal tap. He had a stroke. He was still mobile but was dissatisfied with his recovery. He now has a huge tumour growing in his spine that's rendered him a quadriplegic requiring 24-hour nursing care. He was brave enough to come forward and share his story, as were his family. I fear many others aren't. Closer to home, and for those of you who'd like to listen to the podcast, it's still available on the ABC Radio National website in background briefing. But I think it's a chilling example here of the death of a woman called Sheila Drysdale, who had advanced dementia, who was in a nursing home, whose husband had had treatment for his sore knees by a cosmetic surgeon in Sydney. And he did some research after he'd had treatment. He'd first heard about the treatment on the radio in an ad. He did some treatment, Googled a group in America, although they operate in the Bahamas, and they said, of course, we treat dementia all the time, bring her over. He couldn't do that. It was too expensive, too difficult because of her condition. So he went back and petitioned the doctor who treated him and said, can you help my wife? And the doctor did. Never seen the patient before, treated the patient the same day. The husband gave consent. The patient died within 7 hours of the treatment. She bled to death back in the nursing home. Now, the coroner's report, which Anne Arnold covers in this article and which we also covered in a paper we wrote recently, talks about a cluster of errors. So, yes, it was bad that the doctor didn't ask and inappropriately didn't ask about whether she was on any blood thinning reagents. It was bad that back in the nursing home, the nurses didn't, or the staff didn't treat a woman who'd had liposuction appropriately post-operatively. Yes, it was bad that the doctor discharged the patient after she'd fainted in his suite. But why was she there in the first place? They thought she was having treatment for dementia. Now, that's fundamentally wrong. And I think this is a quote directly from the coroner, that it was quack medicine. So, what to look for? These are my warning signs. If it's not part of a clinical trial, think twice. If it's the same treatment being offered for many conditions, including erectile dysfunction and baldness, think twice. If they're asking for payment, think twice. If they claim benefits based on testimonials, if they're saying that there's no harm because the cells come from you, so it's natural, think twice. If your specialist doesn't know about it, I think that should definitely ring some alarm bells. So thank you very much, and I look forward to questions.
Thanks so much, Megan. That was just fascinating. I have to say I personally didn't know a lot about stem cells before your presentation, but I know a lot more now, and I'm also slightly alarmed by some of the things you said. But the best thing is, I think the fact that alerting everyone to potential misinformation is just so important. So I found your presentation very enlightening. If anyone has any questions, could I ask that you make your way to either microphone, which is located in the aisles on either side of the seating. As I said before, I encourage you to be brave and ask a question. I'm sure some people have got some burning questions. I've certainly got a couple of questions myself, so I'll give people a moment to actually make their way to the microphone if they have a question that they have been they've formed during Megan's presentation. So, Megan, one thing that I was sort of interested in, you mentioned about the Therapeutic Goods Administration and how important that is with some of the providers of these stem cell treatments. I'm interested as to whether the legislation is actually keeping up with the clinical research, given that's your area of interest and expertise.
So thanks, Jen. I think it is, if it's being done right. So there's definitely a framework and support and steps that are in place that will and do allow for good research to happen. If we have a promising approach that we want to trial on patients, we can apply to get to participate in a clinical trial. We can go to an ethics committee and have to justify what we're doing and meet lots of other standards. But what I think is concerning is that even though they've been alerted to this issue, there's been no action. And there's some concern. And I understand it to some extent. They don't want to suppress a promising area, because I get a bit carried away because I'm so angry and cranky about the kind of exploitative practices. But there may be benefit in some of these interventions. But we just need to know and ask more questions. And I don't think we're ready at the stage yet where we should just let it go, let it rip. The TGA have a role here. But as does the Australian Health Practitioners Regulatory Agency around conduct of doctors. As does ACCC in advertising. So there's many bodies that should be coming together. So I think it works well when it's being used appropriately. but it's not responsive to deviations. And I think these clinics have been able to say that what they're doing is just medicine. It doesn't require, it's not a product, so therefore it doesn't require TGA.
Anella, a question.
So, Megan, maybe it's worth also discussing the difference between a clinical trial conducted in a robust way with ethics approval and trials that happen within those clinics that are kind of market research for the clinic and pass themselves off as being good clinical practice in trials, and how maybe consumers can work out what the difference is in that.
Well, I think you've raised an excellent point, and I think that the case study in Florida where the women were rendered blind is exactly that. Again, making assumptions. The NIH is like, you know, the premier American body. So when I still do use their clinical trials registry, I assumed that there's a whole lot of vetting that sat alongside or occurred before a clinical trial was posted, and information about a clinical trial. Because I just showed you the top layer, if you like, of a search. But if we clicked on any of those trials that I showed you, under that is a lot of information about, you know, more details about what the clinical trial involves, how many patients are being registered, but also how to participate. So that's great if you use it properly, because it means that patients can work out if there's a clinical trial near them. That's what it was designed for. But what I didn't realise, there's no vetting. I can contact them, and in fact a colleague of mine actually did, made up a fictitious clinical trial, submitted it, and it got posted. So that's concerning, right? So you make an assumption about what's happening. So this clinic in Florida, I think they pretended they were doing a clinical trial. I think they had in-house ethics approval. So let's talk about ethics approval. So in Australia, animal research and research involving humans, whether it's taking a biopsy of a cell or you participating in our sociology study, we have to go to our local ethics committee and we have to make a case. We have to say, we want to work with these animals or these humans because we want to answer this question. And we've thought about this, and we know that there's some issues involved in what we're doing, and perhaps there's some harms or risks, and this is how we're going to address them. And if the committee doesn't think we've done it well enough, they'll tell us. They'll say, you can't do this without addressing this. If we fail to do appropriate or request appropriate approval, we are unable to publish our research, because often when those papers I've shown you, if they involve humans, I have to declare who my ethics committee was and who's approved my project and put the code in of my project. Also, if we're funded by government, we have to have ethics committee approval. Now, I think all that's absolutely fabulous, but how can it still be abused? Well, if you have your own Mickey Mouse ethics committee and no one's checking, then no one knows, I suppose. So there's those groups that are completely fictitious and who are just sort of, if you like, co-opting and using science jargon and terminology. In fact, one of the Australian websites had a quote, which is an absolute favourite, a statement on their website that said, once we have treated enough patients to show safety and efficacy, basically to show it's safe and that it works, we're then going to do a clinical trial. And so I'm thinking, kind of... the other area that I think Ornella's alluding to, which I think is also worth talking about, is a case study. So there is another layer sort of further down. It's not a clinical trial, but I suppose is a formal registration of patients who have had treatment. And it gets in a grey zone. So these are also ethically reviewed and ethically reviewed by legitimate committees. But I feel like it's still a little bit grey because patients are paying. And what we're seeing in a few cases in Australia is clinics that are doing great clinical research that we really, really want to see more of, but kind of subsidising it with a sideline of selling it. And the justification is that it's too expensive to do clinical trials, and it is expensive. But I feel like there's some profiteering going on as well. So it's a really difficult one. I don't have the answer. It's expensive and I want to see it go on, but I feel like there's no incentive to do this if you can already do this. So I don't know if that answers your question, but it's concerning.
Hello, and thank you very much for your talk. Fascinating. What about issues around cerebral palsy and Parkinson's? Is there any prospect of treatment for those conditions in the future, or where do we stand?
Okay, so fantastic questions, and these are certainly areas that are highly promoted by these clinics. I'll take Parkinson's first. So there's actually been decades of work using stem cells in clinical trials for Parkinson's disease. In Parkinson's disease, there's a discrete cluster of nerves in the brain that are damaged. And the idea is, using stem cells, that we can make those damaged nerves, or if you like, the precursor or early nerves, and put them back exactly into that area and make them basically fill the gap. And what groups in Sweden and other countries around in Europe did some decades ago, or a decade or so ago, was to do exactly that. They used fetal cells, though. And they used fetal cells, and they put the cells, the nerves made from fetal cells, into the brain. And what they observed, and that was only in a handful of patients, so it was one of those early phase 1, phase 2 studies. What they observed, and they monitored these patients for many, many years, is that some of the patients had benefit, but some of the patients had exacerbation of symptoms. So their symptoms got worse and some got better. So that raises a really interesting conundrum about what went wrong. It's great some people got better. They have also now, some of those patients have passed away and donated their brains to research, and they've been able to show that those cells are still alive, or still not alive, just poor phrasing, sorry, that they did actually were still present in the brain. So it showed that they could stay there for a long time, which is good if we're trying to deliver a cell therapy. So it started to answer some great questions, but it raised many more. And I think that's what we're going to see in this field, is that at this early stage, we're going to raise more questions than we have answers. So what's exciting about Parkinson's disease is that there's new approaches are being undertaken. So they're learning from those early studies, and they're trying to work out how to make the right type of stem cells in the right number. The right type of cells from stem cells in the right number. And colleagues of ours here at the Florey in Melbourne are doing some wonderful work in animal studies where they're doing that sort of preclinical research, showing how to make those cells and put them back into the brain of animals at scale. They are also partnering with groups who are doing the next generation human clinical trials, and we're hoping that they will be making an announcement and starting a clinical trial next year. It will probably be overseas initially, but then coming to Australia. These will again be early phase clinical trials, and there will be strict requirements around who is enrolled, but we want to learn again more. So we're at a stage now, having had that initial discovery, gone away and sort of perfected the way to make these cells. We're now at a stage where we're ready to start trialling them on patients again. With cerebral palsy, it's a little bit more complex, I think, because what's happened in cerebral palsy, I think it depends very much on the child, and it's very broad. There are clinical trials being undertaken overseas on very young children with cerebral palsy, where cells are being added back into the fluid around the brain to see whether those cells can help in either preventing further neural damage and further behavioural problems for the children. But those studies haven't finished their analysis. However, participants in those studies have hit the airwaves in America. So I've seen an amazing video on like Today program or something like that, where a little boy was filmed by his parents, quite debilitated, had the cord blood in this case, transplantation, and then was looking great and could shoot little hoops in a basketball ring. Absolutely fantastic for that boy. Was it the stem cells? Was he always going to improve? We don't know. And I think with a condition like cerebral palsy, there's many cells that are affected. It's not that discrete cluster like Parkinson's disease. So often when I answer this question about what conditions will be the first ones helped by stem cells, I think it will be those conditions where there's a discrete type of cell that's affected, where we can make those cells and put them back into the patient. I'm not sure whether we can do something effectively at scale across a whole organ, like a whole kidney or a whole brain.
Megan, I was going to ask you, often people are talking about having a plasma-rich protein, PRP, and feel that they've really had stem cell treatment. And I just wondered if you could make a comment about that.
Sure. So PRP, platelet-rich plasma, is marketed heavily, and many of the clinics, the 77 clinics I mentioned in Australia, offer both. So platelet-rich plasma is basically taking a sample of your blood from your vein, so peripheral blood. There aren't usually any circulating stem cells in your blood. They reside in your bone marrow. But they take the blood, they centrifuge it, and then they collect a fraction of that and put it back into you. Some clinics just offer that. Again, because it's using the patient's own cells, there's no TGA requirement. There's been, to my understanding, very little evidence collected associated with the benefit of that treatment, but it's highly promoted. It's about $600, maybe a bit more. And I've certainly got family members who've had hip problems, knee problems, who've had the treatment, and often keep coming back to have the treatment. So one of the things I'd raise is the serum making a difference at all, or is it just the injection that's making a difference? So is the cells at all? So having said that that treatment, PRP, doesn't involve stem cells, it's still marketed as stem cells. And it's a great point to raise actually, that those 77 clinics that I mentioned, I bet there's not stem cells in the stuff that goes back into the body in most of them. But they use that term. So I think it's a buzzword.
But I think often people will often say they've actually had stem cell therapy, and it can cost $600, but sometimes $1,500 or $2,000, and it's just the confusion around the two different procedures and the terminology.
And again, I think the things that I'd be considering are similar to the warning signs I raised. I think it's a conversation you have with your specialist as well. And just because the clinic's promoting it doesn't necessarily make them specialists in that area. Yeah. Of course, it becomes more difficult if it's a specialist who's offering the treatment, but I'd be getting a second opinion.
Would you all please join with me?
In closing, I also want to thank Megan for such an informative talk. Every time I sit with Megan and have meetings, I just wanted to keep going because it's just a fascinating topic, lots of issues and just lots of things to think about. So, and thank you for everybody for coming out tonight. It's a woolly weather out there, and I really appreciate you coming, so thank you.